oncology is advancing at a very rapid rate. And with all advancements, it comes with its challenges. We're being asked to look at more and more markers on ever diminishing sizes of sample. We're seeing turnaround times increase. And unfortunately, although we're getting much more information for many patients, we're getting no information on some. And is that a reasonable compromise? But maybe it isn't a compromise at all. It's my pleasure to introduce Dr. Darren Davis today to talk to us about how we can overcome that compromise and get the best of both worlds. I met Darren, 12, 15 years ago, Darren developed a circulating tumour cell enrichment and isolation technology called ApoCell, which is an area I was working in at the time. But of course, developing technologies is an expensive business, so he also built a service business on the side so that he could fund that work. And then 10, 15 years later, here we are again, working with Darren, but this time on the service business side. That business was ultimately bought by Precision for Medicine, and Darren's the Senior Vice President at Precision for Medicine. So welcome, Darren.
Yeah, good afternoon. Thank you, Daryl, and thank you to Agena for putting together this symposium. It's a great pleasure to be able to share some exciting data that we generated today in our Houston-based laboratory. As Daryl was mentioning, I founded the laboratory in 2005 to support biomarkers in clinical trials, using a variety of technologies to be able to help drive solutions with our clients. And we continue to do that today through innovation and advancement in our workflows in the laboratory to help create novel solutions. So I would like to start out by presenting a little bit about Precision Medicine Group, which some of you may not be familiar with. And essentially, the founders of Precision for Medicine Group started about 12 years ago. And the vision was really to create an entity that covered the drug development life cycle. So that included everything from preclinical drug development to supporting biomarkers in clinical trials, having the regulatory support to advance those biomarkers through the regulatory agencies, and then commercialization of those drugs or diagnostics that are approved to be able to expand market access to the patients around the world. So essentially, Precision for Medicine Group consists of two business units. One is Precision for Medicine, that's where I fall under. The other is Precision AQ. And Precision for Medicine really focuses on the clinical trial side. We have a CRO group that helps find patients to put on these rare trials. And on the other side is Precision AQ with the commercialization, where we have investment analyst firm look at market access data and helping the clients achieve greater indications for their approved drugs or diagnostics. So just to give you a little sense of how fast we've grown over the last 12 years, it's quite impressive. We have about 6 specialty labs, 2 in Europe, or actually 1 in Europe, 1 in the UK, and then the rest are in the US. That's indicated by the yellow dots. It's a little bit small to read. But importantly, we also have laboratories around the world to support processing of fresh blood samples. So that's indicated by the purple dots. So we represent every major continent or region to be able to get those samples quickly to the processing lab where they're stabilized. And then we also have a global presence with offices around the world. So right now we're probably about 3,600 plus employees. We continue to grow quite rapidly. The founders closed a deal with Blackstone in 2021, and so that private equity group is putting more cash into the business, and we continue to grow both organically and through acquisitions. So just a little bit more in depth about where the Houston-based lab fits in and how we ended up working with Agena. So as I mentioned, we focus on the clinical trial spectrum, and we're able to offer these services a la carte or with a comprehensive solution supporting clinical trials and laboratory translational solutions. And we also, in that effort, provide project managers to serve as a single point of contact and really support every spectrum of the drug development life cycle. So that could be just research projects, it could be advancing technologies, looking at different workflows. And that's really why Precision is coined a specialty service lab as opposed to a general laboratory. And importantly, we also have the ability to manage data sciences. We have a group that is a separate business that Quartz Bio is able to manage a lot of clinical data as well as laboratory data. And importantly, in this whole effort, we also provide central lab services to be able to get the samples from the site to the laboratory in a timely manner and stabilized manner. So as Daryl mentioned, the ApoStream technology, which is our proprietary CTC technology, I'm not going to talk about that today, but we do have a special validated kit that's required to really maintain stability of the blood sample during transit so that we can process that sample reliably once it reaches the laboratory in Houston. And then finally, we have our CDX capability. So we have a compliant facility in Frederick, Maryland, where we can support single-site PMAs. So we've taken CDXs all the way through from development through the regulatory agency to the market. So a little bit about our experience in tissue analysis. So we are considered a specialty lab, and one of those areas is our ability to work with tissues. And we have everything from what I would call your traditional histopathology processing, which includes embedding biopsies that we receive, special stains, performing immunohistochemistry, and importantly, we have board-certified pathologists on staff that can enable reading of the samples. And on this context, we also have global studies that we can support processing of samples in Europe with centralized reading in the US. Actually, one of the key features of our specialty business around tissues is our ability to develop multiplex immunofluorescent panels. This is actually how I established the laboratory in 2005 with the support of Pfizer, AstraZeneca, and Novartis. In this context, we've advanced the technology quite a bit these days, so we're now using up to 60-plex panels for our discovery process, all the way through to 8-plex panels more in the clinical trial stage. And these panels are validated to the different levels from fit for purpose to CLIA to be able to support some of these trials and patient enrollment into the studies. We also have made significant investments in our digital pathology infrastructure, which includes the ability to be able to perform centralized readings through 21 CFR Part 11 controlled software, along with AI analysis on the back end. And we also have a variety of platforms to support molecular analysis of tissues. Obviously, the Agena mass array is one. We do a lot of ddPCR, PCR, and NGS sequencing as well. So in this context, to be able to support tissue-based studies, we also built a biorepository. This biorepository is located in Winston-Salem, and our medical director there manages the biorepository with over 3 million FFP samples covering through from oncology through other diseases. And it's very, very comprehensive. We use these tissues for assay development, validation, or research studies with our clients. And essentially, we can provide these samples to our clients for use at their own labs, but we also use them internally to develop our own assays and validation for clinical studies. Or in the context of this study I'm going to present shortly here, we were able to actually use a lot of our specimens that we've characterized previously through sequencing. So one of our ongoing efforts is really to continue to build annotated inventory for the pharmaceutical biotech industry to be able to use these samples in their own capacity for validation, as well as our own needs internally. So we have over 10,000 samples sequenced to date. This is an ongoing program that we continue to implement, where we use the Alchemine precision assay, the Thermo Fisher platform or the Illumina platform using the TSO 500. So the slide, the image on the left shows kind of the breadth of samples that we have sequenced to date. So over 23 different types of cancer. And on the right shows an example of the types of tissues and the variants that we've found in lung cancer, including ROS1 fusions and EGFR variants. So now shifting gears, obviously that was a bit of a stage to set up the project that we conducted using the mass array system in our laboratory in Houston, Texas. And for those of you that aren't familiar with this technology, as Daryl mentioned, I first came across it about 10 or 12 years ago, looking at it as a solution for a liquid biopsy CTC test. But essentially, we've brought it on to the laboratory in Houston to help look at these samples that have failed sequencing. And essentially, this technology allows you to do tens to hundreds of genetic targets to look at multiplexing simultaneously. It's very impressive with its turnaround time in the sense that it can take 8 to 10 hours. And I think one of the best parts of this assay is that it's very cost effective with respect to other sequencing technologies. We often have challenges supporting sequencing for clinical trials with limited budgets. So this technology offers another solution. And essentially, for those of you that aren't familiar with it, it's a proprietary MALDI-TOF variant detection system with essentially lasers irradiate the DNA molecules and obliterate them where they're sucked up in a vacuum towards the top, and then a detector senses those particles. And the beauty about this system is that the output or data analysis is actually done automatically, as shown there in the top right-hand corner, looking at the spectrum. So just to compare a little bit about sequencing, a little bit further in depth, shown on the right side, some pros and cons with sequencing versus using the mass array, as well as the workflow. So the workflow, very similar to PCR. We can take the extracted material, look at variants enriched, basically using the Siegel base extension. And again, this is a very fast turnaround time. So in a setting like Precision for Medicine Lab, we have some clients that want fast turnaround times. So 1 to 2 days versus 10 to 14 days with sequencing makes a huge difference, especially in the clinical trial context where our clients are looking for rapid data delivery. And essentially, when the data acquisition is done, the system automatically generates the output. So that's something else that is very beneficial in our environment. We don't have to outsource the data to third-party bioinformatics, or sometimes we do extensive analysis and base space. So this just presents another alternative solution for those complicated projects. And essentially, you know, the amazing thing about this is its ability to identify samples that have previously failed sequencing. So in general, those rates can reach up to 22% by NGS, and with the mass array, we tend to see about 2% of the ability to detect the mutant variants. So just to kind of highlight this study that we conducted, looking at samples that previously failed NGS, either extraction or sequencing, we wanted to see, basically evaluate these samples using the iPlex HS Lung version 2 panel, and essentially that looks at 89 hotspot variants across 5 genes, the most common genes, and essentially we took 196 lung tumor biopsy samples that had previously failed QNS. 89 of those were by the TSO 500 and 107 from the Oncomine precision assay. So these samples I mentioned basically failed either at extraction phase or after sequencing. And all samples were processed using the mass array Agena iPlex Pro Sample ID, which is a QC step to test DNA integrity, and then subsequently with the Agena lung version 2 panel. So this slide summarizes the samples and the sequencing quality or quantity where they failed. So shown in the blue are the number of samples that failed TSO 500, and shown in the purple are the number of samples that failed OPA. So you can see on the left-hand side that quite a few samples failed. 109 failed the DNA QC step at extraction. And then after sequencing, the other 84 failed either on the Thermo Fisher platform or the Illumina platform. So all of these samples were then put through the Agena QC step, and they passed the system and actually passed the ability to detect variants. And that data is summarized here. So pretty fascinating results. Out of the 196 samples, we could see that 38% of the samples, we could find actual variants. So somewhat surprising to us, and remarkable sensitivity, I would say here. And not only that, but the distribution of these variants are shown on the left-hand side in the table. Gives you an idea of the sensitivity and depth of the types of variants, but we're able to find variants in all of the 5 genes that the panel is designed to detect. And importantly, the distribution of these mutations is about what we'd expect in the literature, KRAS and EGFR being the most prevalent. So we also performed orthogonal validation in this context to ensure that we had reliable detection of the variants, and so we used ddPCR. And in this experiment, we used basically what Bio-Rad had available off the shelf to look at these specific variants that were available, KRAS, EGFR, exon 19 deletions, and BRAF, some of them. And interestingly, that we found 100% positive predictive value and 99.7% negative predictive value. And that was the 2 samples that basically, it was around the KRAS mutation where ddPCR was able to pick it up. And then again, this is going through the whole workflow and having an automated output of the data analysis. And as we looked further into the system, we could actually find that those samples were indeed positive, but not reported through the automated manner. So that kind of really sums up the pilot project that we conducted with our samples from our biorepository that failed sequencing. And so in summary, the Agena iPlex HS lung panel and the mass array system is highly tolerant of poor QNS FFPE samples. And this really enabled us to recover and deliver accurate results that otherwise would have failed NGS-based analysis.
Heard about solutions with tissue testing and how we can quicken the turnaround time and get rid of that 3-letter word of QNS. But there are some tissue challenges that we can't overcome with technological advances in tissue. And that is, well, if we can't safely get a tissue biopsy, then there's nothing we can do with a tissue technology analysis, can we? Well, I'm sure everyone agrees with that. But also a tissue biopsy is only a small piece of one possible nodule, not really always representative of the entire global disease. And lastly, tissue biopsies are generally only done at one point in time and not frequently over the course of disease. And these are all opportunities for liquid biopsy to complement highly robust tissue analysis, and I'm going to talk to you about that today. We have a second chemistry that runs on the same instrument, and that is our Ultraseq chemistry that we use for tissue analysis. And this is a short amplicon PCR because, again, liquid biopsy is degraded, but it's reliably degraded, unlike FFPE, that's unreliably degraded, but it is degraded. So short amplicon PCR and a single PCR reaction. because we're looking down to 0.1% minor allele frequency, or 1 in 1,000. And we don't want to do multiple PCR reactions because you don't have that amount of material from a liquid biopsy. So a single PCR reaction, recommending 10 to 20 nanograms of input, which is 3 to 6,000 molecules, and at 0.1%, that's 3 to 6 molecules of the variant. We have targeted panels, and again, as you heard before, simple, automated, user-friendly analysis. So this is the chemistry here, very similar to what you saw Darren present before, which is short amplicon PCR followed by single base extension, but this time our terminator nucleotide is labelled with biotin, such that we can pull that out with a streptavidin labelled bead, washing away any unused primer and other material, and only having the mutant product that's present that drives our sensitivity down that order of magnitude that's required to move from tissue to liquid biopsy. Now, just some examples. This is a well-known mutation, EGFR T790M. This is a titration of control material. You can see 2% variant allele frequency, or going all the way down to 0.125% variant allele frequency. You can see those peaks very clearly, but what you can also see is the ratio to that other peak to the left of it. That is one of our controls, and that is how we quantify the variant allele frequency. Because of course, if we've only terminated on the mutant and washed everything else away, we need to quantify, and that's our capability of quantifying the variant allele frequency that is present. We have a variety of different panels, but we also have our custom assay development group, which we call Assays by Agena. So we have panels, a very focused EGFR panel, T790M, the main driver mutations, and C797S. And we have our broader lung panel, which looks at, again, 5 genes, more than 70 relevant mutations. We have a panel for colon, and we also have a panel for melanoma, and there are some others that exist as well. So I want to go through a series of studies and show you how the UltraSeq chemistry and panels work. So this first one was done by AstraZeneca, published a couple of years back. The reference is in front of you, but also on the next slide. It was a head-to-head technology comparison of a variety of different technologies. They were testing limits of detection from 20% all the way down to 0.5% across things like qPCR, the UltraSeq mass array, next generation sequencing, digital droplet PCR, and for some reason Sanger and CE. Anyway, so this is the comparison, and the UltraSeq technology was one of the top performers. In fact, it was the UltraSeq technology and the Oncomine that were the two top performers in being able to detect those low frequency mutations down to that 0.5% minor allele frequency. So we're showing here good limits of detection, detecting those rare variants in a liquid biopsy setting. But what about precision? If we want to move into treatment monitoring, we can't just detect, we need to be precise on the variant allele frequency. So with the the Cancer ID Consortium over in Europe, we teamed up with them to do, again, this was another technology comparison study, looking at the precision of the quantitative measurement. And again, comparing here digital PCR, the mass array UltraSeq, as well as next generation sequencing technologies. And I'm just going to focus right in the middle there. That is the CV on the quantitative measurement. Right? So the mass array was at a 9% intraplate and 10% interplate CV, which was the best of all of the technologies compared. And this is using SeroCare reference material. Right, so we've got accuracy and we've got precision. So let's move into treatment monitoring. Now, I've been at sessions this morning talking about molecular clearance, molecular residual disease, that's looking at where you get to, you can't see it anymore, but we wanted to see if we could see earlier than that. So what we wanted to measure was molecular dynamics, and that is taking a sample at time point 0, which is before therapy, taking another sample 4 weeks into therapy, but not looking to see whether it's visible or not visible, but whether it's actually in the trajectory of getting to not visible. Is it going down in frequency, requiring that precision in the medicine, in the measurement? So we've got 38 NSCLC patients here collected at time point 0 and time point 4 weeks. We did also get another sample at progression, which is another question, but I'm focused on what is the outcome if the tumour frequency is dropping at 4 weeks. And the threshold we set was 15% reduction in the variant allele frequency or not, right? So 15% or more reduction versus not going down 15%, staying stable or going up. We just had that very simple classifier. So we measured these samples, and you can see down the bottom here the Kaplan-Meier plots for progression-free survival. If you were reducing, then it was a median 46 weeks progression-free survival versus 6 weeks if you were not dropping. And over on the overall survival, if you were dropping at that 4-week time point, then your overall survival was 145 weeks average versus 30 weeks average. So significant differences, 6 to 40 versus 46 weeks on progression. Just under a year to 3 years on overall survival. And those that we saw that were dropping, 64% had a durable clinical benefit, and that is they remained reduced for at least 6 months, and no observance of the disease coming back in this particular study. All of these are published, by the way, and those references are down the bottom there. So moving on to another topic in my pet area, as you heard, Darren and I worked on CTCs a long time ago. So this is an analysis of circulating tumour cells in melanoma. So here what we've done is we've taken circulating tumour cells and a variety of circulating tumour cells from patients both pre and post-diagnosis, and we have isolated the single cell. So now what we're doing is we're putting a single cell into our reaction and amplifying that single cell. So that's 2 copies, 6.6 picograms of input, and with one of our off-the-shelf panels. So you can see our limit of input down to 1 cell, 6.6 picograms, this is what makes us highly robust, but also allows us to look at a single cell without preamplification. Of course, in a circulating tumour cell, I can use 2 copies, because I only need to see a 50% minor allele frequency in a cell, because it's only got 2 copies, right? So therefore, you can see that math that we're using over on the liquid biopsy. It's not a limitation of the technology. Can we analyse a smaller amount of sample? Yes, of course we can analyse a smaller amount of sample. We can analyse 2 molecules. But you're not going to see a 0.1% mutation frequency when you only put 2 copies of DNA in, right? So anyway, to expand on this particular study, we are looking at those single circulating tumour cells and the profiles of mutations that are in those single circulating tumour cells. And we saw significant heterogeneity in the profiles of those single cells from the same, multiple cells from the same patient before therapy, and particularly after therapy. Melanoma, of course, large number have BRAF V600, largely V600E before therapy, but we saw circulating tumour cells, V600E plus another mutation, for example, NRAS or so on and so forth, or cells that didn't have V600E but did have an NRAS mutation, as an example. So we saw that heterogeneity. And then after therapy, we saw also significant heterogeneity, a significant reduction in the V600E if they had it beforehand, because that defined the therapy that they were on and the therapy that was giving pressure to their disease. But we saw that really broad heterogeneity after, explaining some of these resistance mechanisms and the possible future recurrence outcomes. So lastly, I want to jump track from mutations in liquid biopsy to methylation in liquid biopsy. Well, this is sort of liquid biopsy that I'm presenting here, and this is work of another group. We were not authors on this particular study. So we're looking at CLL, so chronic lymphocytic leukemia, and generating a methylation profile. Well, with single-base extension chemistry, when you do a bisulfite conversion, you can do a single-base extension on that C to T transition, that is the methylated cytosine in the CpG dinucleotide, and you can measure the frequency of that. And they developed a panel of multiple gene, multiple markers to look at risk and progression in chronic lymphocytic leukemia. 2 different studies shown to you down the bottom there. Very clear differences between the 2, what they've called methylation IPLEX types. So that was the first study on bisulfite conversion. I'm not a particularly big fan of bisulfite conversion, especially when we get from, this is a blood cancer study where you get pretty good quality material. When you move over to cell-free DNA, it becomes a lot trickier. And we wanted to move into cell-free DNA because we'd been funded as part of the European Medicines Association to do a large collaborative study in breast cancer. So this is neoadjuvant treatment monitoring in breast cancer. So it's not advanced disease, it's in the neoadjuvant setting. And we developed a panel of methylation markers, 14 different genes, and to get away from bisulfite conversion, we've used methylation-specific restriction enzymes. And then with the ability of our technology to count the number of molecules, then we're working out what the count of the number of molecules is, taking samples at multiple time points over the course of time. And we were partnered with Philips Imaging, one of the very large imaging companies out there who were using their latest, just released imaging to see if our liquid biopsy markers could improve the imaging. And down on the right-hand side there, you can see that yes, it did. That red, and a little bit small for you to see there, I'm sorry, but the red is with the methylation markers and the blue is without. So we did see an improvement in the treatment monitoring and the ultimate prediction of outcomes by incorporating these methylation marks in the neoadjuvant setting of breast cancer. So we've talked today about liquid biopsy, tumour profiling. You did hear a little bit about our sample integrity and sample validity from Darren earlier, and I've spoken about methylation. There are a variety of other applications that are possible on the platform. We have a session coming up directly after this in pharmacogenomics, and I do encourage you to stay on because we're going to be talking about oncology pharmacogenomics as well as other applications of pharmacogenomics. But Dr. Vicky Bratt, who is the lead author on AMP's recommendations for DPYD and 5FU therapy, will be presenting those new recommendations. So I encourage you to think about the other side of oncology, the pharmacogenetics side. So please do stay on, and a variety of other applications, infectious disease, chimerism, blood typing, and others available on the technology on the same box. So the mass array technology, multiplexed analysis of 10 to hundreds of targets from a minimal amount of sample. You know, getting down to 2 input copies, and then from there we just work the math in order to get to your desired limit of detection. Cost-effective analysis, $10 to $100 per sample, depending on the panel and the size. An efficient 1 to 2 day workflow, and customizable applications. Plenty of kits on the shelf, but also a team of 20 scientists that develop custom assays day in, day out, and partner with you to deliver exactly what you want, instead of us forcing what we think you want on you.